Good morning. My name is Lucia Gutierrez, and I'm an associate professor at the University of Wisconsin-Madison. And today I'm going to talk about genomic selection in cereals. But first, I'd like to thank Elizabeth, Mark, and all the organizers at Cornell for the invitation. So, and we're going to start with some breeding history. You can see throughout history, we have had tremendous grain yield increases in maize production. And this is a consequence of the main breeding targets, so breeding and also management. In terms of breeding targets, the main breeding target was yield, and the ultimate goal was food. But today we're facing new challenges. So we have a growing population that we're going to need to feed. We have higher climate variability and other environmental impacts of agriculture that we need to address and manage. Consumers are demanding higher quality and healthier food. And also, we need to make sure that we produce all this food, maintaining the livelihood of the farmers and having communities that are thriving. So all of this poses more challenges to breeding efforts, and we need to be more efficient, have more precision, and make the breeding faster. So for making this happen, I'm going to start with the breeding equation, which is this. And the breeding equation, or how much response do we get from selection, is a function of the intensity of selection. So how many individuals do we select out of the total population, the accuracy with which we evaluate those individuals, the genetic variance or the square root of that, and then the time that it takes to go from one cycle or complete one full cycle. My talk today is going to be focused on line development. So this is for inbred species or hybrid species that use line development to produce hybrids. And I'm going to separate two concepts, and we're going to walk through the talk in two different components. So one is the population improvement, which is the selection of the parents, the intermating, and then the advancement of those individuals until we make it into the next parents. And so this is just one of the main goals of breeding, is to improve that population from which we're going to be deriving individuals. And then for release, we need to derive those lines, and those could be inbred lines, or we are developing those inbred lines to produce a hybrid. So those cultivar development is the second phase here on the right, where we use multiple year evaluation, multiple environments. We do selection for individuals now, not for breeding values, and then we release individuals. Of course, we are cycling. All of this is not disconnected. They are all completely connected. And we're cycling individuals back as parents. So we're using different strategies for selecting those parents. And one aspect that we can change is in this breeding equation is time, right? How much does it take to complete a full generation until we can recycle the parents back? And for that, there have been several strategies that have worked. Some is double haploids that have worked in many species. And today I'm going to talk a little bit about speed breeding, which is another strategy that helps in decreasing the time that it requires to complete a full cycle. So in this case, it means the speed breeding is nothing else than just manipulating the light, the photoperiod, and the temperature together with the nutrients, so that we can make the plants flower faster, so that we can make them shorter times. And in this example, what we did is we evaluated those speed breeding in oats, and we found that we could go from 111 days to harvest until 79. So that means that we can increase and put a few more generations in a year when we are doing this. In our context, we're using it as a single seed descent strategy so that we can get fixed lines faster. But there are other ways in which we can use this speed breeding. So this is one way that we can increase the response to selection, is by decreasing the time. How fast can we recycle these parents? Well, we're still evaluating a few options. You can go as fast as the S2, which would be very similar to animal breeding. So these concepts come from animal breeding before. Hickey has been working a lot on this population improvement and speed and cultivar development. We can go as fast as we want. It depends on a few things. Our training population, how are we going to be updating those, and then our ability to screen those individuals. How many genotypes can we score in those? And then a few other concepts. We talked about the variance, how much the genetic variance will affect the response to selection. So one way that breeders have always worked is we never do crosses that are just the good by the good. We also look for different traits and complementarity of traits. And so we can use the same strategies when we're looking at genomic selection. We can look at the cross instead of the parents. Instead of selecting the best parents, we select the best crosses. That could potentially produce a higher variance. That could produce a higher top 10% of the individuals. So these are some strategies that we can use to increase the response to selection. And this follows the work by the group in Minnesota with Kevin Smith and others that have done this software to do this cross-selection. Other ways that we can improve, the response to selection is by increasing the accuracy. And we can play with the accuracy in many ways. And you've probably seen, and there's going to be other talks that talk about models and a few other things to do to improve the accuracy. Here I'm going to focus on optimizing the training population. So this is our main hypothesis was if we have a large number of individuals that were genotyped and phenotyped, and they are, part of our breeding program, so they are mildly related to the testing individual, to the individuals we would like to predict. And our question was, so are we better off by using all the individuals in the training population, or are we better off by selecting a subset that is going to be more predictive of those individuals that we would like to predict on the testing? And so here what I have is, this is the predictive ability, so the correlation between the observed and the predicted breeding values. And this is the number of individuals in the x-axis. And you can see here is the full population. So if we use all the individuals in this population, this is the predictive ability we're going to get for predicting new individuals. And so what we did is we evaluated a few strategies. Some of them are like predictor error of variance, CD means, and a few others that have been published. And then we proposed this new strategy that is based on weighting a stratified sample of the individuals based on the relationship to the testing, so that we make sure that we always have a highly related individual of the testing in the training. And then also if we have some structure in that population, that we are weighting them by those groups. So in that way, what we call this WC or weighted kinship matrix, what we found is that we had a higher predictive ability. We could find an optimal here training population for those individuals that was higher than the random sample and then the variance of a lot of different random samples. It was also better than the CD mean and the PEV. So these are strategies that we can use to improve the predictive ability if we know what individuals we would like to test. Other things that we need to take into account when we're reading and we're trying to use as a genomic selection is how to optimize the training population for the environment. So the target population of environment is very relevant, and just putting everything together in the same bag might not be the best strategy. So we have shown that using mega environments and predicting within mega environments, or for predicting new environments, it's a good strategy, or just modeling the G by E. If we have the full matrix, then those produce higher predictive ability. So those are common practices in breeding, but we don't need, we need to remember to use those G by E when we do our prediction. We can also use, it's highly recommended to use all the available information. So if we have environmental covariates, we can enrich our predictions. We can increase our predictive ability by using all these environmental covariates. Here are some examples where predictive ability here was increased by using those environmental covariates. Other ways that we can improve the response to selection is by managing the experimental designs. Breeders are very used to doing experimental designs. They're familiar with the way that increasing replication will increase the precision of those means estimates. But then we have done, most of our trials have been using balanced designs across environments. And that's very inefficient. And so what we did is we tried and show a few strategies where we can do is optimize experimental designs first so that we can have a higher predictive ability, but also allocate our resources in a more efficient way by not replicating every single genotype in every single location in the same way, but using our trials, our network of trial as our experiment, and then distributing our genotypes across the trial. If we do that smartly with knowing the megaenvironments, so megaenvironments are these groups of environments that have similar ranking of the genotypes. So if we treat one megaenvironment as one unit, then we can increase our predictive ability. And here I'm showing how we can increase the response to selection by about 40% by using those strategies, using the same number of plots, so the same resources as balancing everything and replicating everything, but now by using this unbalanced designs and not replicating everywhere and treating the megaenvironment as a unit. And this is called megaenvironmental design. So this changes the intensity because we are being able to evaluate more individuals. It also changes the accuracy with which we evaluate those individuals and then the time. So other strategies to improve the response to selection is to improve phenotyping. And this has been booming in the last few years. And there are a lot of strategies at using high throughput phenotyping and other strategies to improve the response to selection. And again, we need very good models. We need very good training data to build those models, but we can improve the phenotyping. If we are trying to predict noise in our training population, then we're going to get noise, right? But if we improve the way we phenotype, then we can increase the response to selection because we're increasing the accuracy. Other strategies that we can use is breeders. We never work with a single trait. We usually have multiple traits that are relevant. Those traits tend to be correlated. And just trying to focus independently on those are usually not the best strategy. But we can use this correlation to improve our predictive ability. And sometimes that can help us predict traits that are very expensive or very hard to evaluate. Some of those are these baking quality traits in bread that are very expensive. And when we do a single trait and we try to predict it, the predictive ability is not very large. But when we use two traits, so a correlated trait to that one, we increase the predictive ability. And this is using a full set. Using all the individuals in the training population phenotype for the second trait. But we can also allocate those which ones do we phenotype for the second trait and for the first one in a smarter way. If we purposefully unbalance those, we can increase the predictive ability having the same sample size as doing one trait at a time. So we can use those multiple traits in a wise way to improve the predictive ability. And then we can also use them in indexes and other ways, or we can use them independently and do independent culling or other strategies that breeders use. This is another example of the replace phenotyping, and this was for molting quality in barley, where again, our goal was to predict some of those molting quality that we do not have the capacity, lab capacity to phenotype all the individuals in our breeding lines. So what happens is that we end up just phenotyping for quality the lines that have passed our agronomic evaluations. And that restricts a lot the diversity from which we can select, and also restricts the progress. But if we can phenotype earlier, because we use correlated traits, or we can predict them based on this training population, then we can predict our early lines, and we can do a better selection and a higher response to selection. So those are some of the strategies that we have used in our program, using genomic selection to improve the efficiency with which we do the breeding. I'm going to pass an advertisement here. So there is going to be a session in the agronomy meetings that are going to happen in November, where we're going to discuss some of the designs for predictive agriculture. So I invite all of you to attend and encourage you to submit. And then with that, I'd like to thank you and thank the organizers for the invitation, and I'll be happy to take questions.